many diseases can be successfully treated in the simple environment of a cell culture dish. But to successfully treat real people, the drug agent has to take a journey through the infinitely more complex environment within our bodies and arrive intact inside the affected cells. This process, called drug delivery, is one of the most significant barriers in medicine. A collaboration between Lawrence Berkeley National Laboratory and Genentech, a member of the Roche Group, is working to break through some of the drug delivery bottlenecks by designing the most effective lipid nanoparticles (LNPs), tiny spherical pouches made of fatty molecules that encapsulate therapeutic agents until they dock with cell membranes and release their contents. The first drug to use LNPs was approved in the year 2018, but the delivery method rose to global prominence with the Pfizer and Moderna mRNA COVID vaccines. The researcher says that it is quite a smart system because if you just deliver the RNA itself to the human body, the RNA is degraded by nucleases and cannot easily cross the cell membrane due to its size and charge. But the LNPs deliver it safely into the cell. LNPs are now being widely explored as a delivery system for vaccines for other infectious diseases or therapeutic vaccines for cancer. The viability of these new applications will be dependent on how well the lipid envelopes fuse with target cells, how stable the drug LNP formulations are in storage so that they have a long shelf life, and how stable they are in the body so they can confer prolonged drug activity. All these properties are controlled by the mixture of molecules used to create the LNP and the resulting 3D structure of the particle. The research team has been studying how to tune the structure of LNPs for desired properties for several years. Their latest paper, published in the journal ACS Nano, documents how a high-throughput workflow allows them to produce and characterize LNPs at record speed. The study also includes the first-ever demonstration of how LNP structure correlates with the activity of its contents, which for this investigation was an antisense oligonucleotide (ASO). ASOs are small snippets of RNA or DNA base pairs that block gene expression by binding to strands of mRNA and preventing them from being translated into proteins. ASOs are a great way to treat diseases caused by faulty proteins or the overabundance of a protein. But like mRNA, they are susceptible to roving nucleases, enzymes that degrade RNA and DNA, and cells do not readily uptake them. The scientists discovered that ASO carrying LNPs with neatly ordered, closely packed internal structures led to better silencing of a faulty gene in human neurons that is associated with a degenerative disease compared with LNPs that had a more disordered structure. The findings were from cell-based studies, not from animal studies. So there is still more work ahead, but the team are excited to build upon these insights using the complementary tools of each institution.